So hello and welcome to my presentation, which is on the effect of CytoD on Vasa accumulation during Drosophila Oogenesis. So I'm a senior at Hopkins. I'm a mole cell bio major, and I work in Tatjana Trcek's research lab. And there we focus on germ granules, which for those who may not know, are a non-membrane bound organelle, and they're located in the posterior pole of Drosophila embryos, which, as the name suggests, is where the germ cells develop. And these granules specifically are an interesting field because they are what it's looking like right now, associated with rapid processing of mRNAs, which lead to the development of those germ cells. So to give a little background, Vasa mRNAs are known components of germ granules, and so therefore they're the easiest way for us to visualize granule quantity and location using GFP tags. So we have already used Vasa GFP image analysis to formulate this little model here for the quantity of germ cells present as oocytes develop into embryos. So it really comes in 3 stages that you can see. During oogenesis, there's an accumulation of the Vasa mRNA, which again is the proxy for the germ granules. During embryogenesis, there's mostly this flatline persistence phase. And then during the maternal to zygotic transition, we see a degradation of those Vasa mRNA and thus the germ granules. So we wanted to further understand the mechanisms of our granule formation now that we have this model by perturbing it and understanding the effects of those changes. So in this experiment, we use CytoD to do just that on a pharmacological level. Jumping ahead a little bit here in the video. So in normal oogenesis, the somatic nurse cells, which you'll see around here, provide nutrients to the oocyte until the end of stage 10, which is when they go through programmed cell death, and that allows all of their nutrients and the space that they formerly occupied to be consumed by this growing oocyte. And so we can see that here in the video. These are our nurse cells. This is the oocyte. And so this process is called nurse cell dumping. CytoD has been shown to inhibit actin polymerization and therefore prevent nurse cell dumping, since we can see over here in this is a model of the stage development of Drosophila oocytes. The nurse cell dumping process is heavily dependent on actin filaments. So this CytoD inhibition also inhibits that dumping process. And since we know that nurse cells dump some of the other mRNAs that help form germ granules, including nanos, which we saw on the last slide, we hypothesized that inhibiting dumping would prevent the formation of new granules as measured by our Vasa GFP image analysis. So I performed the computational analysis of these GFP results in ImageJ with a new procedure that we developed to overcome a background fluorescence issue that had invalidated past experiments. So in the past, we would just take a larger image like this of the entire posterior half of the oocyte, but the problem arose with the background fluorescence up here, separate from the germplasm, which was shifting the numbers a little bit. So in this new method, we go through 3 important steps, which are first, that the images, which are 3D images, are imported into ImageJ and then cropped around the germplasm, as you can see right here in figure 1. Then the 3D image counter, which is a feature in ImageJ, was used to measure the integrated density of the GFP fluorescence. So we can see that in figure 2. That's kind of what the object counter looks like. And then again, we're measuring integrated density, which is the best metric for fluorescence. And then finally in figure 3, the different part of this method is that the threshold set on the object counter is dynamic. So I would adjust the slider bar until that there is no background fluorescence being picked up and there are no large clumps present in the germplasm that we are analyzing. Finally, for data analysis, again, integrated density metric was used, and then it was graphed on a percentage y-axis, where the highest integrated density value from the data set became the numerator, and all the other points were a fraction relative to that highest value. And that allows us to look more on a grand scale at the shape of the accumulation rather than just plainly the metrics themselves. And kind of the easiest way to explain that is just to show you. So we'll dive into our first control, which were the untreated control. So as you can see in the graph here, the highest value is often one of the last, since these are graphs of accumulation. So we set the second to last one here as 100%, and all the other data points are given as a fraction relative to that 100%. The images up here that you see were captured at slightly different time points. So they are supposed to represent each of the beginnings of 3 different phases that we see here. So A is reflective of the beginning of the experiment. As you can see, this looks like a normal stage 10 Drosophila embryo. Around point B is when we can see dumping begin, and that's visibly reflected in the image itself, and then graphically on the steep increase in accumulation. And then the last part is the plateau. Once the nurse cells seem to be mostly or all gone there, the accumulation returns to just a gradual slope, and the image itself is visibly different. So all of the images analyzed were captured at slightly different starting time points. So I normalized their x-axes to the beginning of dumping, since that's such a visible thing in the images themselves. And then the quantitative highlights that I pulled out of this data set are that dumping begins around 190 minutes, and the start of the plateau phase occurs at 370 minutes, and at that point, 73% of the total accumulation had occurred. So we can compare that first set of data with our further control set, which subjected the embryos to only the vehicle that was being used to deliver CytoD, but not the drug itself. So that allows us to make sure that that vehicle doesn't have any unintended effect on our controls. The images again confirm that dumping was successful. We see again our stage 10, the dumping and the completion, and we had very similar time points, similar graphical shape and similar time points, with dumping beginning again right around the 200-minute mark, the plateau occurring around the 400-minute mark, and the total accumulation at about 75%. In the experimental data set, however, we can immediately tell from the images that there's no sign of dumping. So these all look like stage 10. We didn't see any of the expansion of the oocyte or the retraction of the nurse cells that we saw in the previous control sets. And then I pulled these 3 images from obligatory time points, which is why they're labeled 1, 2, and 3 instead of A, B, and C, because I wanted to emphasize that they're no longer correlated to the dumping or plateau phases as the last ones were, since we didn't see either of those phases occur. So from these results, we're able to easily conclude that the CytoD treatment did have the effect of preventing nurse cell dumping and subsequently prevented the rapid new germ granule formation during the accumulation phase of oocyte development. The other really important takeaway from this was the new method of image analysis and the consistency that we saw in the controls, which matched what we would expect from the other wild type research that we've done to create the models, indicates that the analysis method was accurate and successful. And that's the most important result because the CytoD was fairly well studied, but this method will be very useful to us in the future when we plan to utilize it in genetic perturbation experiments, which should be a lot cleaner and more decisive than the pharmacological experiments that we've done with CytoD. And that is it. So I will take any questions now. And I'll take a look at the chat too. Yes, these were Z stacks, Moira. And so the ImageJ runs the integrated density for each of those layers all at once. So the threshold, the best way to do it is one of those Z stacks is the point where the cell is best in focus. So that's going to have the brightest fluorescence. So that's really what's determining everything else, but it does run through all the other layers to make sure that everything is getting picked up. So that we use that brightest layer to set the threshold.
Thank you. All right, any other questions for Dylan? Okay, with that, thank you so much, Dylan. That was a great presentation.